Good morning, everybody. I'm going to get started so we leave plenty of time for our speaker today. This morning I have the pleasure of introducing our speaker, Dr. Phil Greenberg, who, as many of you I'm sure know, is a professor of medicine and immunology here at the University of Washington. He is the head of the program in immunology in the clinical research division at the Fred Hutch. At the beginning of his training, he did his medical residency and postdoctoral research at UC San Diego. He then completed his oncology fellowship at the Fred Hutch and joined the UW faculty, and he has been an academic leader here in Seattle for over 40 years. His research has focused on elucidating the principles underlying interactions between T cells and tumors, and then developing cellular and molecular approaches to manipulate T cell immunity to promote tumor eradication. He has won numerous honors and awards, I assure you a small sampling of which include selection as an investigator for the Parker Institute for Cancer Immunotherapy. He's been elected to the American Society of Clinical Investigation, the Association of American Physicians, the American College of Physicians, the American Association for the Advancement of Science. That is quite an alphabet soup. He is on the board of directors for the American Association of Cancer Research, and he is a distinguished fellow of the American Association of Immunologists. And just this year, in 2019, he received the American Society of Hematology's E. Donald Thomas Prize for Pioneering Research Achievements in Hematology. Dr. Greenberg has published well over 200 articles. He is on the editorial board of over 10 journals, including his ongoing service as the editor-in-chief of Cancer Immunology Research. And finally, and I think quite remarkably, he remains a vigorous educator here at UW. He continues to mentor young researchers, and he teaches several immunology courses. So please join me in welcoming Dr. Greenberg to the podium.
Thanks very much. So I think I have to get, is my slide next? Got it. Okay. Yeah. Well, thanks very much, and I appreciate all the rest of you also got up early to come here this morning. This isn't going to be a global history. It's actually going to be a bit more of a personal history of cancer immunotherapy. It's been a long road for us. We've been trying to develop effective T cell therapies for malignancies, and I think we're finally really getting close, and have had some at least good clinical signals already. So this isn't exactly what the place looked like when I came. It was a little bit earlier, but it actually, sadly, isn't that far off. I think I actually went into the original, the RR tower that got built shortly after this. So we've all seen enormous changes here, and so has the field, and it's been a wonderful evolution and a joy to be a part of. So I'm going to just start out with a brief history here of where my lab has come from. As you can imagine, a career in science is not an individual effort. It takes a village. And I had the joy of actually having two wonderful mentors, actually, two remarkable scientists, Don Thomas, who recruited me here, and Alex Pfeffer, who gave me the opportunity to work in his lab and develop my own independent projects. Don, it was an interesting issue when I interviewed with Don. He had already been starting to develop bone marrow transplantation. And it was just around that was working in a basic immunology lab, and we were looking at the way T cells recognize targets, and it was around that time that it became clear that in bone marrow transplantation, a very significant part of the anti-leukemic effect, and often the cures, were dependent on a graft versus host effect that delivered a graft versus leukemia activity, demonstrating for the first time really in humans that T cells were capable of eradicating tumors. And so in an interview with Don, I said to him, Don, honestly, I think this bone marrow transplant regimen is just way too toxic, and I think we can replace it with T cell therapy, and you won't have to do bone marrow transplantation anymore. And Don, being Don, kind of looked at me a bit askance and asked me if I really thought I could do that, and I said, oh, of course. Now, Don actually still went on and asked me to join the faculty. Now, to make it clear, Don went on and got the Nobel Prize for bone marrow transplantation. In 2012, we celebrated the millionth bone marrow transplant, or hematopoietic stem cell transplant. So it didn't exactly disappear very quickly, and I don't think Don actually really worried that I was about to replace him. But when I started, I had this vision that we could actually develop T-cell therapy for cancer. And during those early years, in the late 70s to early 80s, we started really working entirely only in mouse models. And Mac Cheever was a colleague, and it was a small lab. It was just myself, actually, Mac Cheever, and Alex Pfeffer, who ran the lab. Those who look at me during this year often call these my rabbinical years. But it was an exciting time, and whoops, I have to go back. We started actually just looking at what were the principles, what is required to make immune cells recognize cancer, how does that operate? Again, all in mouse models. And we started then the initial effort to see whether we could expand cells outside the body, create an immune response, and then put it back in to actually treat a cancer. When I set up my own lab, we still were using mouse models with the idea of building that towards translation. And I was very fortunate because it's very hard when you start a young lab to get trainees who believe in projects and it really was a blessing. And I had some really remarkable young people at the time. And this is when we really started, growth factors were being cloned, we started using them, we developed ways of actually cloning individual T cells outside the body. We started understanding how T cells respond to signals and grow, and then also began to start looking at this problem of, if you have a T cell that recognizes a tumor, why doesn't it get rid of it all the time? And this issue of CD8 T cell tolerance became quite important, and we started trying to understand that and how that worked. And of course, that's grown into this enormous field now of T cell exhaustion and dysfunction and energy. As we were advancing with our mouse work, in the 1990s, several physician scientists came into my lab, and they were much more impatient than I was about moving things out of the lab into the clinic. And in particular, Stan Riddell was critical in essentially demanding that we start trying to apply this to people. And so we did. And we, again, then had to learn how to work with human T cells and tissue culture in order to essentially, again, build immune responses outside the body that you could then put into the body and make it work. And we realized that what we needed was a target that was unequivocal. And so the first studies we did were proof of principle in that we would treat a viral disease, and we would actually use the T cells to recognize and prevent virus. And we showed that we could prevent CMV infection in patients after bone marrow transplantation by providing them with T cell clones that were specific to CMV. It was the first evidence of antigen-specific T cell immune reconstitution in people. We also started actually, even in the 1990s, toying with the idea that we could genetically modify cells. And when we looked at treating HIV, we performed really the first study of genetically modified antigen-specific T cells into people. And we started now trying to also understand what is it that makes a T cell when you manipulate it ex vivo and then put it into a patient, what is it that's required for that cell to persist and function and maintain an immune response after you transfer it. Around the same time, we realized that there was a whole lot more work to be done in terms of genetic engineering. And although we were at the tip of the iceberg, we realized there was much more work to do. And a group of people who came in the lab really created they were risk takers, and they really created a whole new set of possibilities for us. And this is again with Stan Riddell, who was actually helped pushing this, but actually Mike Jensen came in the lab and in the 1990s started developing CAR T cells actually, and then took that project and actually has used that to run his own lab now, which is now based again. He's returned to Seattle again, and he's now at Children's Hospital. And Stan has also now essentially taken the idea of CAR T cells and run with that. And there were a whole other group of investigators who were also helping us understand how could we do genetic manipulation. And there was a lot to be done, but we now started understanding how we could introduce receptor genes in the cells so that we could impart specificity to cells, so you could very rapidly create essentially a whole army of cells with a defined specificity. We understood the importance of co-stimulation, which has been critical both to the development of CAR T cells as well as T cells, and that we could genetically engineer cells to do that. And we even began, at the time, starting to look at editing T cells. So this was before there was the CRISPR-Cas9 systems we have now, but this was back in the time when the reagents were really zinc finger nucleases. But we started showing that we could remove T cells, remove endogenous T cell receptors, and replace those genes with our own set of genes. And this has all become very important for where the field is moving now. Of course, after the proof of principle in viruses, we needed to show that what we initially set out to do was that we could treat human malignancies. And that took a small group of people who were actually very committed to that. And in particular, Cassian Yu came in the lab, and his project became actually to develop T cell therapy for melanoma, because we had evidence that we had antigens in melanoma that we could target. And indeed, Cassian published the first paper showing that antigen-specific T cell clones could target a malignancy, that they would localize to the tumor, that they would eradicate the tumor. And we then started looking again for now additional targets outside of melanoma, recognizing that there was this whole world of malignancies that was going to need to be approached. And also, how can we isolate more efficiently antigen-specific T cells and develop a method that's actually now in wide use. During that same window, another group of people came in the lab, and they really decided to expand our horizons, essentially start looking beyond these initial efforts into viruses and what we knew were highly antigenic malignancies. And this was a time when we started looking at targeting things like lymphomas. We did a study showing we could treat leukemia with antigen-specific T cell clones. And we even actually looked at this combination of checkpoint therapy, anti-CTLA-4 blockade with T cells, based in part on works of a very good friend and collaborator, Jim Allison, who had been developing that, and as you know, is an approved therapy now, as we'll talk about in a few minutes. We also started trying to understand how can we specifically track the fate of individual T cells. We didn't quite yet have the high-dimensional analysis I'll show you we have now, but it was our initial foray into this. But there was still a lot of basic science that needed to get done. And so there was a group of people in the lab who came in and really started looking at new ideas and basically allowing these people to work and pursue projects really, again, changed the directions that the lab could go in. And during this time, we started looking again at what is making T cells dysfunctional in the context of a tumor. How do you rescue cells from that dysfunction? In addition, we started looking at what other tumors could we go after. And Ingen, as I'll show you later, developed this model for treating pancreatic cancer that we will be in the clinic with in 2020. So this is now what the lab looks like now. It's another generation of scientists. They're quite remarkable. They make going into the lab and interacting very exciting. There's also another part of it. It's funny as you keep looking at the people in your lab, they seem to always be young, and then you look in the mirror and you realize it's not that everybody stays the same age. We all age a whole lot more, but we keep refreshing our lab with really viable, remarkable young people. And this group actually started looking at creating fusion proteins, really more creative ways of doing genetic manipulation. I'll show you a little bit of that. We started playing with how we can make receptors higher affinity. We started getting into single-cell RNA sequencing, and again, looking at T-cell receptor gene therapy as a strategy now for treating malignancy. Now, while we were doing this, a lot else was going on, obviously, and in particular, two colleagues who actually we all did postdocs together had really made some remarkable contributions. Bob Schreiber, who is at this point now at WashU in St. Louis, showed that endogenous T-cell responses really do provide surveillance for tumors and protect us. He really validated the immunosurveillance hypothesis. And Jim Allison, who again, when we were all postdocs in San Diego together, but he was really interested in T-cell activation, what makes a T-cell respond or not respond. And what Jim showed actually was that there weren't just activation signals, there were inhibitory signals that T cells got. and that those inhibitory signals, that balance of activation and inhibition was critical for whether a T cell decided to respond. And he developed a model with CTLA-4. He developed an antibody to CTLA-4, and he showed that if he blocked CTLA-4, you would get immune responses that otherwise you couldn't detect. And indeed, CTLA-4's major role is actually in inducing new responses. It's during the priming phase when T cells are seeing antigen. And as you know, Jim actually, although he was a PhD, pushed this to the clinic, and anti-CTLA-4 was approved as a therapy for treating melanoma initially in 2011. Now, around the same time, or a little bit later actually, Tasuku Hanjo in Japan had cloned a gene, PD-1. He didn't really have a lot of insight into what it was responsible for, but colleagues of his, Gordon, from across the ocean in Boston actually, Gordon Freeman and Arlene Sharp, showed that PD-1 was another inhibitory signal that was being delivered to T cells, and that antibodies to either the receptor PD-1 or the ligand PD-L1, if you block that, you could activate T cells and have them function. And what they showed is this is probably the major mechanism for which T cells get exhausted, or at least was at the time thought to be the dominant mechanism. There are now clearly many other mechanisms, but that blocking anti-PD-1 or PD-L1 would result in clinical effects. And again, PD-1 very rapidly, much more rapidly than anti-CTLA-4, got approved in 2014. Now, what happened shortly after that was people said, well, these are working at different phases of the immune response. CTLA-4 is inducing more responses. PD-1 is working at the effector phase where you're seeing the target. Potentially combining them would be very beneficial. And indeed, it does provide some benefit. And so if you look at what happens, this is in studies in melanoma showing that CTLA-4 alone or PD-1 alone was not as good. And again, it's relatively small differences. You can see these are real. And indeed, this study was actually the pivotal study that resulted in the combination getting approved. Now, the problem, of course, with the combination is that this is blocking not just the response to malignancy, it's blocking the immune responses, these inhibitory signals everywhere in the body. And so autoimmunity is becoming an increasing problem. And so using these in combination still represents a problem because of how much inhibition you're actually taking off, and it really unleashes potential autoimmune responses. But this does cure patients who otherwise have fatal disease. The typical survival curve for melanoma is most patients have expired within 12 months. This is all treating advanced disease. The rest are usually, unfortunately, expire within 24 hours. So these long curves actually was a dramatic difference, and that really was the reason it got approved. The consequence of this was perhaps not surprising. Not this December, but the previous December in 2018, Jim and Tasuku Hanjo were actually awarded the Nobel Prize in Physiology and Medicine. And as I told you, I'm actually quite close friends with Jim for a long time, and I actually was with him when the award was announced. It was an interesting issue. It gets announced at 11:30 a.m. in Stockholm. I was with him at a meeting in New York, and I'd been with him several times on the night when we know that it's going to get announced. We've stayed up late many nights. We just wound up drunk in the morning, typically, without much of a call. And again, he didn't get a phone call, which is unusual. Usually you're supposed to get a phone call telling you you're going to get the award before it gets announced. But he didn't, and it was his son, who's actually standing next to him here, who actually was watching CNN while we were all together and saw that it got announced that he got the award at 5:30. And so, as you can tell, by 5:35, we all already had a couple of bottles of champagne, and we were drinking and celebrating. And Jim realized that he was going to have to actually give some conferences, and so he decided he better jump in the shower and get ready. And of course, while he was in the shower was when the call came. So this is Jim accepting the Nobel Prize. He did say yes, of course. And it turned out the reason he didn't get the call in advance was he hadn't realized, but he had changed his cell phone number, and so they had no idea how to reach him. But it does have a happy ending. This is Jim receiving the award in Stockholm a few months later. I did get to celebrate with him. This is Jim Mia, and Louis Lanier, another very close friend. And it was an interesting time. The reason this was at the award ceremony after they received the award, and the king's son actually, king who gives the party actually, his son came over to the three of us. We were standing together, and he said, "I need a picture." He said, "Do the three of you all use the same barber?" So I'll leave it. The answer is no, none of us do, obviously. So, okay, so despite the dramatic benefit you see in some patients, with what I showed you, the checkpoint light, still the majority of patients don't respond. And so we have this enormous achievement, but still we're not going to benefit most patients. And who's responding? Well, it turns out clearly if you sequence tumor and find tumors that have high mutational burdens, that the T cells are largely seeing new antigens caused by mutations. So high mutations predict responses, but that still wasn't enough. And actually looking at the tumor tissue has been very instructive. So the people who are responding have high mutational burdens, but they also have this kind of phenotype where this is the tumor and green is T cells here, and the T cells have highly invaded the tumor. But now they've stopped functioning, and that's one of the reasons why if it expresses PD-L1 and you can block it, you can unleash these responses and get good therapeutic activity. But that's not how most tumors look, unfortunately. Most tumors are either, more commonly have this kind of picture where you do see some T cells, but you can see they're being excluded from the tumor, and the tumor has ways of keeping T cells out, and that's one of the ways of turning off an effective immune response. And so if you can disrupt the architecture of the tumor microenvironment, you can actually modify that for that group of patients. But having said that, just the same, it's still, there are all these patients where there's no T cells to be seen. And so the problem is, how do we address this? And this is really where I think the strategy we and others have pursued, which is that you're going to have to create a response for these patients. You're going to have to provide a T cell that actually can see the tumor. And now that we can engineer T cells, this really makes a difference. So, I mean, as you know, CAR T cells have had this enormous impact on B-cell malignancies. They've unfortunately had very little success with solid tumors, and there's lots of reasons for that. But one of the reasons is that the receptor for let me back up there. The receptor for a CAR T cell is an antibody ectodomain that's been fused to a set of signaling domains that will activate a T cell. But it's not a very efficient receptor, actually, so you need lots and lots of that antigen expressed on the target for this to actually deliver a signal. A T cell receptor, by contrast, is a much more sensitive receptor. It can see very low antigen density, but the magnitude of the signal it provides from seeing very low density is much less, much weaker than the signal that comes through a CAR. So CAR T cells tend to expand dramatically if they see if they're actually seeing enough of their target. T cells don't expand at that same rate, and so that is an issue. And so we need to work on that to engineer a T cell so it will respond better through its TCR. But it really exposes us to a lot more targets. Because this has an antibody ectodomain, it can only see surface proteins. TCRs see these small peptides that are derived from inside the cell and put on the cell surface, and are much more efficient at actually, and have a much broader set of targets. And since most of the antigens that get changed in malignancy are internal, not surface proteins, it really opens up a whole realm of being able to target malignancy. I'm just showing you, this was again the studies that led to the approval of CAR T cells. These were patients who had literally weeks to months to live. The overall survival was actually quite dramatic. This was a study of 75 patients done at Penn. And in the group, there were 61 of the 75 patients who achieved complete remissions. As you can see, these responses are actually quite durable. Many of these patients presumably are cured, unfortunately not all of them, but it does tell us that this really can provide a durable, lasting response, and people have now resolved what are the reasons for failure, what is the reason why responses are not durable, et cetera. But that did lead to the approval of CAR T cells, originally targeting CD19 and ALL in 2017. Now, for our lab, we were actually still trying to find targets for TCRs. And we used initially AML as a model malignancy to target, in part because of our history and because of some foolish comments I had made to Don Thomas actually several decades ago. And so the initial screen was to just analyze differential gene expression in leukemia versus normal cells. This protein WT1, which is an oncogene, fell out. It was originally described in Japan by Sugiyama. And if you stain for protein in malignancies, you can see the vast majority of AMLs are expressing this at logarithmically higher levels than are found in any normal cell. So tumors are co-opting this and using it to help drive the malignancy. Now, in leukemia, although the stem cell hypothesis is somewhat arguable for many epithelial tumors, it's not arguable for leukemia. We can detect by animal transplantation the primordial stem cell that really drives leukemia. And so we asked, is WT1 expressed in that cell? Because obviously getting rid of the leukemic stem cell is critical for actually providing benefit. And we collaborated with Ravi Majetti and Irv Weissman at Stanford, who had developed a technique for isolating leukemic stem cells. And we looked, where is WT1 expressed in leukemic stem cells? And this is the stem cell group, and it's expressed at very high levels, as you can see in red. And this is in normal hematopoietic stem cells. It is present, but at very low levels. And again, they're separated by logs. Now, we also needed to know safety. Is it found in normal tissues? And we looked at all genes that were differentially expressed in leukemia and leukemic stem cells and hematopoietic stem cells. And then also, are they expressed in normal tissues? And only 7 genes really fell out that looked like they might be targetable. One of them, fortunately, is WT1. It is expressed in a somatic cell. It's found in these supporting cells, these podocytes in the kidney and the glomerulus. It seems to be an immunologically privileged site. We've never been able to get a T cell to enter into the glomerulus supporting structure, and so a toxicity has never been seen targeting WT1 for that. But WT1 did fall out. There was one other gene that fell out, and I'm going to show you that now because I'm going to come back to it for a brief moment later. Was a gene called cyclin A1, or CCNA1 is the gene nomenclature. It's not a gene that's actually expressed in somatic cells. It's an obligate gene for meiosis, so it's expressed in sperm and ova. It's not, but it turns out that a variety of malignancies, including leukemia, co-opt this and actually use it for mitosis. And so the only place where we found it was in the testes in actually sperm. And so this looks like an interesting target as well, and I'll come back to that, because it seems to have a reciprocal relationship actually with WT1. We next needed to isolate a high-affinity T-cell receptor for WT1, since that's what we chose to target, and test it. And I'm going to show you the results of a clinical trial that we very recently published. We had first done, as I alluded to earlier, a proof of principle where we isolated WT1-specific CD8 T-cell clones from bone marrow donors, expanded them, gave them to the patient, and showed that we had anti-leukemic activity. The problem is cloning out T-cells from single cell to 10 billion is actually a very labor-intensive effort and very time-consuming effort, and not a practical clinical strategy for therapy, but a good way to prove a principle that a target is a viable target. And so we did a trial in leukemia post-transplant again. These were patients who were at very high risk of relapse. We learned in that first trial with the clones that T cells were going to have a hard time keeping up with rapidly progressing leukemia. We sadly saw that again. But that they could actually treat minimal residual disease or prevent relapse at patients at high risk. So the trial we designed was initially to prevent relapse in patients who had very high risk. I'll show you what I mean by that in a moment. But of course, because we were enrolling patients pre-transplant, some of the patients who were at very high risk turned out to be at very, very high risk and actually relapsed before we could actually give them the cells. And so we did also have a treatment arm, and I'll show you some of that data as well, because we've learned a lot from that. So this is what I mean by poor risk. Many of these patients were already, it was secondary AML, it was their second transplant, they had poor cytogenetics, they had extramedullary disease. And this is comparing, the dark is the patients we enrolled, the orange are patients who were at the Hutch at the same time, but because they didn't express HLA-A2, which was the MHC allele we were targeting with the TCR, they couldn't be on the trial. And so that's the control group. And if you look at the matched controls, and we had to censor out deaths that occurred before we were ready to treat patients, and we waited about 60 to 90 days to actually give them T cells because we needed to make sure that graft versus host disease had dissipated. We censored out any deaths that occurred before that in the matched controls, and so the relapse rate is about 40%. It actually otherwise is actually quite a bit higher, but I think what was impressive was this. So this is what happened in the patients who got the T cells. So match controls, the p-value even for 12 patients here is a p less than 0.01. This was published in Nature Med a few months ago. And again, this is looking at the relapses, or this is a swimmer's plot. We haven't updated this. This was what appeared in the paper. But the median follow-up now is more than 48 months. The likelihood of relapsing, particularly with high-risk disease at that point, is not exactly, but it's very close to zero. So the likelihood that these patients didn't receive benefit is, as you see, is less than 0.01. Now we need to understand why are these cells working in this setting, why are they not working in other settings, and to do that, we started now getting into high-dimensional analysis using single-cell RNA-seq. And this is just one of the approaches that we've used. We take viable single-cell suspensions. This is a 10x technology where single cells get put into a bead with a droplet that contains the primers for creating libraries. Each gene gets its own ID, its own unique marker. Each transcript gets a different gets identified, and you can identify individual genes and individual cells this way, and then you can plot them using these high-dimensional graphic strategies. This is a Tisney plot. I'll show you another one as well a bit later. And each of these colors represents a distinct cell population that are separated based on the fact that not because you know anything about them except what genes they express, and so they're put on the plot based on their similar gene expression. And then you can look at what genes are differentially expressed, and you do the analysis. And a lot of this in our group has been done by Dan Hunter in Ochupui's lab. So this is essentially a patient who we treated with T cells on that trial, who remained in remission at a year. More than 10% of the cells that made up his CD8 compartment were the cells we gave. So these cells were persisting quite long. We wanted to look at what was how did they differ genetically from the cells we had given the patient. So this is looking at it in 3 dimensions, because this has been reduced to 2 dimensions. This is in 3 dimensions. And now what I'm marking here in blue are only those T cells that actually have the TCR gene we put in. So you can distinguish them from the other cells based on that gene. And of course, you can tell the graphical presentation to show any gene that you're interested in looking at. So if you do that, what you can see is that the T cells are really embedded completely in this, mostly in the CD8 effector memory population. Some are in the central memory population. And I'm not going to show you the data. What we showed is that almost all of the cells we were finding in the effector memory and effector population were derived from central memory cells based on the fact that they expressed the same TCR gene, and so they represented cells that differentiated from central memory to effector memory. Now, based on this trial, which at least looks very promising in prophylaxis, we now have a non-transplant trial that's open, and what we're looking at now is trying to prevent relapse again in patients who undergo induction therapy, and with a variety of excellent collaborators right now. And this trial really is AML patients, again, at high risk. And again, the high risk is defined by if they get induction therapy, which is a very aggressive chemotherapy, and they don't achieve complete remission. So they either have minimal residual disease after their first induction, or just have incomplete marrow recovery, which has turned out to be evidence, actually, of leukemia in the bone marrow. That's why their marrow is not fully recovering, even though you can't necessarily detect it. So we've taken those patients, and I'll show you, this is what their prognosis looks like, so that more than 80% of those patients are going to relapse in the first year, no matter what you do after that induction therapy. So you give them a lot more therapy, you can get a large fraction into remission, but it doesn't matter. They're destined, unfortunately, to relapse in that first year. So the plan is actually all those patients are recommended to go to transplant, but there are a lot of patients who aren't candidates for transplant, in particular elderly patients. And so our study right now is mostly in an elderly population. And so this is where we are right now. We've treated 7 patients. I'll show you the data. So these are patients who, when we treated them with T cells, had no evidence of disease. If it's green here, these patients are now out more than a year. These 2 patients and remain in complete remission. This is a patient who actually had minimal residual disease when we gave the T cells. The minimal residual disease disappeared. Patient remains in complete remission. This is another patient who had minimal residual disease. We gave the T cells. Patient ultimately became what looks to be in complete remission, but then did relapse back at home, decided she didn't want to come back to Seattle for more therapy, and unfortunately has expired. And then there's another patient, again, with minimal residual disease we treated. The minimal residual disease disappeared. The T cells remained, and the patient remains in complete remission again. There are 2 patients here who we did treat who relapsed. One of them we know relapsed with a WT1 negative leukemia, again suggesting that T cells are probably selecting out antigen loss variant, as I'll show you, unfortunately could happen. And there's another patient who, again, didn't achieve a complete remission, and this one actually looks more like this failure of T cells to persist as the leukemia progressed, and I'll talk about that in a moment too. Now, as I said, we had a second arm to this, which was to treat those patients who did relapse. And the problem here is, although we saw activity, it wasn't good enough. There are 12 patients who relapsed after transplant. We have some evidence. This is one of the patients. The red is the blast that was seen in the patient's marrow when we gave the T cells arrows of the T cell infusions. You can see the leukemia disappeared. That was quite encouraging. He wasn't receiving any more therapy. But this patient ultimately, unfortunately, went on and relapsed about a year later after that. So activity, but not good enough. Again, the treatment arm versus the control group. If anything, the prognostic factors for the treatment arm were worse than the patients who were in the control group. And what we saw was, again, evidence very suggestive of anti-leukemic activity. So we saw early prolongation of survival. But I think the unfortunate message here is that when we looked at long-term survival, we weren't really impacting that. Now, we know that the major obstacle here is that T cells really are unable to persist in most patients with rapidly progressing leukemia, and so we need to do something more in order to make T cells able to persist when leukemia is growing. Myeloid cells in particular are quite immunosuppressive, and that's another problem that has to be broached. Now, this was, of course, disappointing, but it brings back some from one of our history lessons, Teddy Roosevelt, a 26th president, who said nothing worth doing is ever easy, and this certainly has proven not to be easy. Because that's a little different than the attitude of our current president, who actually said, I thought it would be easier. I think this was just before he actually went out to play golf, too, but that's a second story. Okay, so what have we done? We've tried to figure out what's happening here. So we've used single-cell RNA-seq of leukemic cells, again, to reveal mechanisms of escape. And I'm going to just show you a few, not all of the ones that will be appearing in a publication soon. So this is, again, looking at a Tisney plot, and this is all the cells from the bone marrow put into groups. And as you can see, all the blasts, the AML blasts, separate quite well by gene expression from the other populations. And when we looked at the genes that are expressed in those blasts, we saw something that was quite, at the time, very disappointing and surprising. And that, although the way we originally were looking whether patients' leukemia was WT1 positive, was to take a whole population of blasts, do PCR, is WT1 found? And the answer is yes. But when we looked here, and if you can see the orange dots, by single-cell analysis, it was only being expressed in a small percentage. So we had selected out for variants here that no longer express WT1. And we looked at what they do express. And as I told you, we had identified this second gene, cyclin A1 encoded by CCNA1, which was now dramatically upregulated in those leukemic blasts. And we know that there's this reciprocal relationship that in order for leukemia to survive as it loses an oncogene, it needs to co-opt and find a new oncogene to help drive it. And cyclin A1 seems to be a common mechanism by which that occurs. So Rachel Perrett in the lab has now been isolating T-cell receptors that recognize cyclin A1. She found a whole panel of TCRs. This is just looking at their ability to kill fresh leukemic cells. We are now making a GMP vector which will have a cyclin A1 TCR that we can now use, and we can screen patients. Do they have, is their leukemia homogeneous for an antigen? Is it heterogeneous for the two antigens? Should everybody get two TCRs, or should we do them sequentially? These are all answers for another study. So we did have a look at other patients who relapsed. This was one of the patients who was reinduced with chemotherapy and T-cell therapy, but then the AML again rapidly progressed to day 28, despite the fact that the T-cells were in the blood and seemed to be capable of recognizing leukemia. And so we looked at the patient again. This is looking now in the blood, and you can see this small population of AML cells there. And because this was a male-to-donor transplant, we could look at XIST expression in the female cells, because those were the only hematopoietic cells that would exist anymore in the patient. And you can see, so this is the AML. The AML is easy to detect. It expressed WT1, so this wasn't about antigen selection. It expressed HLA-A2, so it hadn't lost the ability to be seen. We had shown another mechanism of escape, immunoproteasome. I won't go through that, but it retained that, so that was not the problem for antigen presentation. So we had to understand what's the problem. Why are these T cells persisting in the blood and yet not working? And I think there's a common message here back from the studies that Jim and Tasco have done. So if you look at, this is a slightly different way of mapping single cells. This is a UMAP plot instead of a Tisney plot, but you see that it separates out cells better, and you can see the CD8 T cells there. And if you look at the cells that express our gene, they're all in this hook off of the CD8 population. And we said, what's special about that? And fortunately now there's ways to not only do RNA analysis at the single cell level, but protein analysis, because it turns out by day 28, when we look for expression of exhaustion markers like PD-1, there wasn't much increase in message. So if we had looked at only single cell RNA-seq, we wouldn't have picked that up. But by staining for protein at the same time, what we found was that there was abundant expression of PD-1 on these cells and not on the other cells in the patient, suggesting that patients, and indeed, had acquired an exhaustion phenotype. This is probably not surprising, considering that the leukemia was rapidly progressing, but it tells us again there's another problem that we need to address to make T cells function in a rapidly progressing leukemia. So we've now looked at a lot of next generation strategies. We need to design better T cells. We need to find ways to make them work. And there's a lot of problems here. We need to have a lot of TCRs. We need to have the cells survive and persist. And we need for them not to become dysfunctional. So we've looked at strategies to identify T cell receptors. I'm not going to go through this. Tom Schmidt in the lab has developed a high throughput technology where we can rapidly, from a whole population of donors, pool out the high affinity TCRs by increased stringency sorting for high affinity T cell receptors. We can express them, we can identify them using a 10x technology and then print them in a DNA printer and create a vector and express them in cells. And what's remarkable now is in about 6 to 10 weeks, we can identify high affinity TCRs that we would be able to advance to the clinic from a panel of donors. So we can get the highest affinity T cell receptor to an antigen that exists in many repertoires very quickly. And this is going to be a really useful strategy for creating more TCRs to take to the clinic. Now that won't solve the problem of that you need a lot more than TCR for a lot of these settings. You need to enhance and sustain T cell function, their anti-tumor activity, and you need to keep the cells alive. And what we know is one of the problems here is there's too many inhibitory signals and not enough costimulatory signals. So how do we address this? So what we decided to do was, when Shannon Oda came in the lab, was to start creating these immunomodulatory fusion proteins that would provide stimulatory signals. Now, you can do better than just provide a stimulatory signal. So this is a TCR complex. But what we can do is we can make these synthetic proteins that, for example, have an ectodomain of an inhibitory receptor, but we've eliminated the cytoplasmic tail, which provides the inhibitory signal, and instead put on it a costimulatory signal. So now, when the malignancy is trying to signal to the T cell, turn yourself off because I'm expressing this inhibitory ligand to contain inflammation here, the T cell actually reads that as an activation signal. So rather than just provide systemic blockade and risk autoimmunity with every cell in the body, we can now create ways of using inhibitory signals to actually drive responses or inhibitory receptors to drive responses rather than turn responses off. And this was one of the leukemia. One of the inhibitory signals it expresses in abundance is CD200. So Shannon made originally, and she published on making a fusion receptor between CD200R and a CD28 signal. And this is just dye dilution. The cells that are moving this way are proliferating. So only the cells that express the dimerizer, the fusion protein, actually were capable of proliferating. They actually kill really well. So this is an assay where you add leukemic cells and T cells, and the T cells actually will eliminate the leukemia. So we compared cells with this IFP versus cells that didn't. So after 24 hours, we added more leukemic targets, and you can see obviously the amount of leukemia, that's what's on this axis, goes up. T cells get rid of it. Both the control T cells and the T cells with the IFP work fine. But by the third exposure to a target, the control T cells are becoming exhausted, dysfunctional, and deleted. And the cells with the IFP actually are capable of eliminating the leukemia again. Now, we showed in a mouse model, this is actually the one that Alex Pfeffer had originally designed with disseminated leukemia. We inject leukemia into mice who wait 5 days. At that point, this is just using IVUS imaging. The leukemia is widely disseminated. We inject Cytoxan and T cells, and then we monitor them for survival. And what you can see is cells with these fusion proteins work much better than cells that lack it. And this is treating relatively minimal disease following the treatment with Cytoxan. So we are advancing these fusion proteins to the clinic, but there is this whole problem of cell death. That cells keep getting activated. The default pathway for T cells is to undergo death. And so the typical pathway, thing that happens is as T cells proliferate and respond, they're relatively insensitive to death early on, but as they become effector cells, they become quite susceptible, and that death is mediated through a death pathway that's mediated by a receptor called FAS. And FAS is a molecule that has an internal cytoplasmic tail that has a death domain, and when its ligand, FAS ligand, binds to it, it trimerizes the receptor, brings the death domains together, and you get the death signal. And so, again, what Shannon rationed was, well, maybe we could just swap that. So now, rather than FAS ligand delivering a death signal, it'll actually deliver a costimulatory signal. Now, because FAS signals as a trimeric molecule, not a dimeric molecule, it turned out, and I'm not going to go with the data, 4,1-BB looks like a much more effective cytoplasmic tail than CD28. Now, FAS ligand is upregulated on leukemic cells and solid tumors because it's interferon responsive. So as a T cell comes in and creates an immune response, it's actually inducing essentially its own death. It even induces FAS ligand on itself, and so T cells in a population actually commit fratricide. So this is a way of trying to address that. We've shown that this actually dramatically improves T cell function. T cells, as they become effector cells, lose their mitochondria. They're incapable of doing fatty acid oxidation. I'm not sure how well it's projecting, but you can see there's a lot more red here in cells that express this IFP. They've upregulated that. We know they're getting a costimulatory signal. I'm not going to show you all that data. But again, if you look at T cell therapy, what you see is that cells with the FAS 4-1BB IFP function much better than cells that lack it. Again, they persist and survive better. Now, the last strategy I'm going to talk about is we need to create a composite interactive T cell response. We know that CD8 T cells like to have CD4 cells interacting with them and providing helper signals and survival signals and growth signals, but it's hard to, but CD4 cells don't typically localize to tumors, particularly tumors that don't express class 2 because CD4 cells are class 2 restricted, CD8s are class 1 restricted. So we know that we can put a receptor into a CD8 T cell. It'll result in activation of the T cell, but as I showed you, they're likely to ultimately become exhausted. So we could try to find a whole new set of targets for CD4 cells, but they still would be unlikely to see epithelial tumors that lack class 2. But what we decided to do was, instead of that, was actually to take a CD4 cell and put the same class 1 restricted T cell receptor in that we are putting into the CD8 T cells. That also simplifies how many genes you have to make. But it turns out that doesn't give you quite enough avidity. You need to actually put in CD8, which also binds to class 1. And so now the CD4 cell that expresses CD8 and the class 1 receptor work better. CD4 cells are also very co-stimulation dependent, so that's probably in the long run not going to be enough, so we have to put in one of these IFPs. That will result in CD4 T cell activation. It will produce IL-2 and help. They also become effector cells and themselves can now directly lyse targets that are expressing class 1. So now we have CD4s and CD8 cells that will wind up in exactly the same environment. And this is just an in vitro study that kind of proves that. This is a class, we put tumor in here at time 0, tumor grows quite rapidly. If we put only the TCR into, this is a WT1 specific TCR into CD4 cells, they have really very little effect in slowing tumor growth. If we add CD8, the T cells do well, they control the tumor, they don't quite get rid of it, but when we add more tumor at 48 hours, we see it go up again, it controls it reasonably well, but won't get rid of it, and again, if we add more. So there's activity, it's not quite enough. It's not as good as what a CD8 T cell does, they're better effector cells. You can see they actually drive the amount of leukemia down, although by the third infusion they're getting exhausted in the absence of CD4 cells. But if we actually put in a one-to-one mix of CD8s and CD4s, the total numbers being the same as we have in these other populations, it's much more effective. You can see they continue to eliminate the leukemia even after the third dose was added. So we're looking at making a composite CD4, CD8 T cell response as well. Now, I just want to talk very briefly about some studies in solid tumors, because that's going to be an important part of where we're going with this. This is our roadmap to treating pancreatic cancer. It was developed by Ingrid Stromness when she was in the lab. She now has her own lab in Minnesota, and these are the postdocs that are currently working on it. And I'm sorry, all of that was done in collaboration with Sunil Lingarani, who is one of the people who really helped develop this mouse model for pancreatic cancer. It's a genetically engineered mouse model, so the oncogenes are knocked into the mouse genome, and they're released in the pancreas by specific Cre recombinase that removes the flux sites. So this is looking at a human tumor and a mouse tumor. These are the pancreatic cancer cells here, but what you see is that most of a pancreatic cancer is not the cancer cells, it's this desmoplastic stroma. There's this very dense fibrosis. And if you look at the mouse tissue and the human tissue, you can't tell the difference unless you've actually looked at whether it's mouse genes or human genes. And so it very well recapitulates the histopathology. It's been used to actually study chemotherapy and develop drugs for pancreatic cancer. It's been very predictive of going from the mouse model to the human for patient responses. The one problem with this model, though, is that because these genes are knocked into the whole genome, ultimately every cell in the pancreas is going to become a cancer. So they don't get one cancer, they keep getting pancreatic cancers. And so that's a bit of a problem for trying to cure. But this is just showing you if you've knocked out P53 and you've expressed KRAS, mice die in about 118 days on median. If you don't knock out P53, it takes a bit longer, and all of those tumors actually endogenously develop a P53 mutation, or virtually all. So we looked at performing adoptive therapy in these mice. We isolated a mesothelin-specific TCR that recognizes the leukemia. Mesothelin is expressed abundantly in the mouse tumor as well as the human tumor, so we picked that because it would model what we could do in patients. We tracked mice with high-resolution ultrasound for the development of tumor, and when they have tumors, we treat them with T cells. We sacrifice them at 8 days or at 28 days, and this is looking at 8 days. So if you look in the spleen at 8 days, if we gave them controlled T cells with an irrelevant TCR, or the ones that have these mesothelin TCR targeting the tumor, in the spleen they're about equivalent. But if you look in the tumor, it's quite different. So these antigen-specific cells are accumulating in the pancreas at the tumor site, whereas the controlled T cells really don't get there with any abundance at all. This is if the red is staining for CD8 T cells. You can see they're abundant if we have an antigen-specific response, and they're actually abutting onto the tumor cells, which are in green here. You see very few T cells in a controlled pancreatic tumor. You said, are they doing anything? Well, as I showed you, there's this very dense stroma in pancreatic cancer. This was looking at 8 days after we gave the cells, so there's a lot of collagen and hyaluronic acid. 8 days later, what you can see is the collagen is almost completely dissolved, so is the hyaluronic acid. Turns out that it's the T cells that when they get in, they secrete gamma interferon. It activates macrophages to make metalloproteinases, which dissolve the stroma. that actually opens up the blood vessels. So this is what blood vessels look like under this dense pressure because of the desmoplastic reaction. You see they're not very patent. This is the same vessels now at 8 days. They're wide open. So this is also a way in which you could deliver things much more effectively to the tumor. So we actually did a randomized study in pancreatic in these mice, where mice were randomized based on ultrasound when they had a tumor to either get relevant T cells or irrelevant T cells. And we treated them every 2 weeks with T cells because we knew that T cells were becoming dysfunctional at about 14 days. Again, this is a problem that needs to be addressed to sustain function. But as you can see, by 12 days later, by ultrasound, the tumor in the pancreas had shrunk. This is a waterfall plot showing that in the majority of mice, we see shrinkage of the initial tumor that we're treating. All of these mice ultimately will progress. And this is just looking at cleaved caspase 3, or apoptosis of tumor cells at the time you have to sacrifice mice. And you can see in mice treated with controlled T cells, tumor cells look actually quite happy. They're progressing quite rapidly. There's a lot of tumor cell death that's continuing to go on in these mice that continue to get treated with T cells, but obviously it's not enough to ultimately rescue them. But as I said, the problem with this model is that all the mice keep getting one tumor after another, and ultimately every pancreatic epithelial cell is destined to become malignant. So again, the p-value here is extraordinary. So we have now advanced this to the clinic. We have isolated a high affinity TCR, again, using Tom Schmidt's strategy in the lab to isolate high affinity TCRs. This is just tetrabinding of those. We have selected the TCR that's going to trial. This is, again, using an assay where we add tumor cells again at 48 hours, and you can see the T cells are controlling tumor growth. Otherwise, the tumor grows actually quite well. This receptor the clinical trial should open around the end of the second quarter, the third quarter of 2020. We will be treating pancreatic cancer with a protocol quite similar to what we had done in the mouse models. Now, that's not the only model we're looking at. We're doing ovarian cancer as well. I'm not going to show you any data on that. These are studies done by Kristen Anderson. This is staining ovarian cancer for mesothelin, the same TCR we're targeting it in pancreatic cancer. It's abundantly overexpressed in the vast majority of patients and quite homogeneous. We're also looking at treating lung cancer. Again, lung cancer overexpresses both WT1 and mesothelin. We're using a genetically engineered model that was created in Tyler Jack's lab. This is Leah Schmidt, who actually did our training. Her graduate work with him is now in my lab, and we're working on developing a therapy where you inject a virus into the lung. It provides Cree, it releases the tumor cells, and what you get here is by 6 weeks you get adenomas, by 16 weeks it's adenocarcinoma. Shortly after that becomes invasive and metastasizes. So we're looking at hopefully by the end of 2020 having clinical trials in a variety of solid tumors. The initial trials will be just for the TCR, but these follow-on trials with further genetic engineering to make cells work better will follow shortly once we've shown safety of targeting the antigens in those settings. So I'm just going to end. Obviously, we can't do any of this without funding. We've been very fortunate. We've applied to conventional and non-conventional funding sources. We found out this, perhaps not so surprising, but we have actually been, we have been remarkably successful and beneficiaries of support from a very large variety of funding sources, including the National Cancer Institute and Stamp to Cancer, and initially from Juno Therapeutics, which is a cell therapy company which was founded in part out of our lab. I've tried to identify the people who were important for this, and so I'm going to stop there. The names are always far too many to give credit to. I hope the pictures actually provided enough insight. Thanks very much.